Welcome. Today, we're diving into the analytical evaluation of transdermal drug delivery systems. This topic explores how drugs are delivered through the skin, offering a promising alternative to traditional methods. We'll look at the science behind it, the challenges, and the potential impact on healthcare. Let's get started. Let's start by exploring transdermal drug delivery systems, or TDDS for short. These are medicated patches that stick to the skin, delivering drugs steadily through intact skin into the bloodstream. This method avoids the digestive system, so the drug isn't broken down before it works. That means more consistent effects, less frequent dosing, and often better patient compliance. Some common examples include nicotine patches for quitting smoking, fentanyl for pain relief, and even patches for Alzheimer's, hormones, and heart conditions. When we talk about global regulatory requirements, there's a lot to keep in mind. In the US, the FDA provides guidance specifically for transdermal and topical delivery systems, including SUPAC guidelines and product-specific guidances. Over in Europe, the EMA focuses on the quality of transdermal patches. Then, we have the ICH guidelines, covering everything from validation and pharmaceutical development to quality risk management and analytical procedures. And don't forget the USP references, which set standards for topical products, drug release, and semi-solid formulations. It's a complex landscape, but understanding these helps us stay compliant worldwide. Let's break down the key qualities we focus on when developing these patches. First, the functional properties, how the drug releases, how well it permeates the skin, and how it sticks. Then, stability attributes, keeping the drug content steady, the adhesive intact, and moisture under control. We call these critical quality attributes, or CQAs. They include physical properties like size, thickness, and weight, plus chemical properties such as the drug assay, impurities, and any leftover solvents, all crucial to ensure safety and effectiveness. Let's talk about how we ensure assay and content uniformity in our samples. We rely on precise analytical techniques like HPLC, UPLC, UV spectroscopy, and LC-MS to get accurate readings. The procedure starts with patch extraction, followed by sample dilution. Then, we run chromatographic analysis and quantify the results against a standard. To meet acceptance criteria, we follow USP guidelines, aiming for 85 to 115% of the label claim, with an acceptance value of 15 or less. Let's walk through an example calculation for a drug assay. This will show us how to apply the concepts we've covered so far in a practical way. We'll break down the steps clearly, so you can see how the numbers come together to give us meaningful results. It's all about turning data into insight and making sure our measurements are accurate and reliable. Let's talk about thickness and weight uniformity. This is all about consistency, making sure lines, strokes, or shapes keep the same visual weight throughout a design. When thickness varies unexpectedly, it can feel off, distracting the eye or breaking the flow. Uniform weight helps create balance and harmony, giving the design a polished, intentional look. It's a subtle detail, but one that makes a big difference in how professional and cohesive something feels. Let's talk about moisture, what it is and why it matters. Moisture content is simply how much water is already in a material. Moisture uptake, on the other hand, is how much water that material can absorb from its environment. Both play a huge role in how materials behave, from strength to durability. Understanding these two concepts helps us predict performance and avoid problems like warping or decay. It's a small detail with big impact. Let's talk about in vitro drug release testing, or IVRT. This is how we evaluate the way a drug releases from its formulation in a controlled lab setting. Common tools include the USP apparatus 5, which uses a paddle over a disc, USB apparatus 6, a rotating cylinder, and the France diffusion cell. We focus on key parameters like the release rate, cumulative release, and release kinetics. This testing is crucial. It supports product development, quality control, and any changes after approval. It's a cornerstone of ensuring consistent, effective medicines. Here, we see a table showing how drug release changes over time. At 1 hour, The release is 120 micrograms per square centimeter. By 2 hours, it's increased to 180. 4 hours in, it reaches 260, and at 6 hours, it's 320. This example helps us understand the rate at which the drug is released experimentally, a key step in evaluating its effectiveness and timing. It's a clear way to track how the drug behaves over several hours. Let's talk about the in vitro skin permeation study, or IVPT. This method helps us understand how substances penetrate the skin. We use different biological membranes, human skin, porcine skin, and synthetic membranes, to simulate real conditions. The France diffusion cell is key here. It measures flux, lag time, and the permeability coefficient, giving us detailed insight into how a compound moves through the skin. This data is crucial for regulatory purposes, especially when proving bioequivalence for generic transdermal drug delivery systems. Let's walk through an example calculation of flux. This will help us see how the concept works in practice, not just in theory. We'll break it down step by step, showing how the flow of a field through a surface is measured. By the end, you'll have a clear sense of how flux connects the math to real-world phenomena. It's a key piece in understanding how fields interact with their surroundings. Let's talk about adhesion performance testing, a crucial step to make sure patches actually stick like they're supposed to. The FDA recommends 3 main tests. First, peel adhesion, measuring the force needed to remove the patch. Then there's the tack test, checking that initial stickiness when you first apply it. And finally, shear adhesion, how well the patch resists sliding or shifting. The goal: the patch stays put for the entire dosing period, with minimal lifting and no sticky residue left behind. This directly impacts how well the therapy works. When we talk about method validation, we're referring to a set of criteria that ensure our testing methods are reliable and accurate. According to ICH guidelines, we focus on specificity, accuracy, precision, linearity, and robustness. These parameters are tested across different aspects like assay, related substances, moisture content, adhesion, and drug release profile. Stability studies are conducted under controlled conditions, long-term, intermediate, and accelerated, to confirm the method holds up over time and varying environments. This gives us confidence in our results. To wrap up, analytical evaluation is key to making sure transdermal drug delivery systems are safe, effective, and high quality. We rely on critical tests like assay, content uniformity, moisture analysis, drug release, skin permeation, and adhesion performance to get a full picture. In vitro release testing and permeation testing are essential tools, both for developing products and meeting regulatory demands. Every analytical method we use must be scientifically justified and validated, following international guidelines. This thorough approach supports the entire product lifecycle, keeps us compliant, and ultimately protects patients. That brings us to the end. For any further support or questions, please don't hesitate to reach out. Thank you all for your time and attention. It's been a pleasure sharing this with you.